cell, okay, the primitive cell, prokaryotic cell, which is the primitive cell. We have the eukaryotic cell, which are the advanced cells, right? And we know that prokaryotic cells do not have any membrane-bound organelles. Eukaryotic cells, on the other hand, have specialized structures. Okay, they have specialized structures and they are complex in function. Okay, they'll be complex in function. Okay, so when we are talking about the structures of the cell, okay, so we have seen in the last class, we have seen a plant cell. A similar will be the animal cell. The only difference with the animal cell is, okay, in the last class, we'll just go back to our, what you're seeing is the central vacuole, okay, but in animal cell we will not find any such vacuole. Okay, we'll not find any such vacuole. We'll have a cell, okay, at the center of the cell we will have the nucleus, and we have this nuclear membrane. Okay, we have the nuclear membrane. All right, then we will have here the chromatin network, then we have the nucleolus, okay, and then we have all the other, compartments or all other structures of the cell suspended in the cytoplasm. So we will have here the cytoplasm, okay, we will have, this is the nucleus. All right, this is outer nuclear membrane, inner nuclear membrane. This is the nucleolus. Then we have the chromatin network. Okay, chromatin network. All right, chromatin network. This chromatin network will form chromosomes. Okay, cytoplasm. This is the endoplasmic reticulum, or the ER. Okay, ER. Then we may have the mitochondria. We may have other vesicular structures. This is mitochondria. Okay, this is mitochondria. So this would be an animal cell. Okay, animal cell. Then we'll have ribosomes, of course. All right, but the basic fact is that in plant cells there is a large central vacuole. A major part of the cell is occupied by that vacuole. Animal cell, we don't see such structure. It's a direct central, the nucleus is placed at the center and the remaining structures are present around. Okay, now when we are talking about cell surface, okay, when we are saying a cell is larger, a cell is smaller, we have different type of cells, okay, different fashioned cells. All right, the cells performing different function. But one important point that we all need to remember is surface to volume ratio. This is very important. Surface to volume ratio. Now, smaller is the cell. Okay, smaller is the cell. It is easy to exchange the material between the cells in and out. If the cells are larger, the exchange becomes little bit difficult, because we are dealing with a larger surface area. But a smaller cell will have smaller surface area. This will enable fast and efficient transport of substances in and out of the cell. What is the speciality of the cell? We see here the cytoplasm, we see here the mitochondria. The cytoplasm, early name of cytoplasm is called as protoplasm. Protoplasm, Purkinje, J.E. Purkinje, he coined the term protoplasm. J.E. Purkinje, he coined the term protoplasm. And Purkinje said that protoplasm is the physical basis of life. That means all important functions, reactions, everything is taking place in the cell. This is what he meant. And definitely, yes, in the cytoplasm, we will see what we call as glucose metabolism. Glucose metabolism. We take in glucose, and this glucose will be broken down to release ATP or energy. Full form of ATP is adenosine triphosphate. Adenosine triphosphate. This is actually the energy currency of the cell. It is the energy currency of the cell. It is the main energy that is utilized by our body to perform various functions. Very important. So, definitely is the physical basis of life, cytoplasm. All major functions are taking place there. In the cytoplasm, we have various organelles that will perform specific function. We're going to study about the different organelles, okay, and how or what functions that they carry out. Now, one important thing that you need to know before we go into the different organelles which are present in the cell, okay, different organelles that are present in the cell. One thing very important that you need to know is that if suppose, for example, let us see here, any cell, consider it a bacteria, a bacteria, consider a plant cell, or consider an animal cell, any of the cell, animal cell, any cell you take, you should always remember that in all of the cell, in their cytoplasm or in their various organelles, there are reactions taking place, and these reactions are called as metabolic reaction. Okay, reactions are taking place inside the cell, and these are specific reactions called metabolism. Like we, in the laboratory, in chemistry, you take up, you take up this test tube, and you use certain chemicals and you carry out reaction. Have you done, Sana? Yes.
Yeah.
Yes, that is a chemical reaction. This is metabolism. This is also chemical reaction. You know what is a chemical reaction?
Sort of.
Yes. Okay, when do you call a reaction as chemical reaction?
When there's like a transformation.
Hmm. Transformation. Heat is given out when there's formation of new compounds, right? So even in metabolism, there is generation of heat, or at times heat may be utilized. There is formation of new compounds. Okay, there's formation of new compounds. So let's see, metabolism. It is defined as the sum total of all chemical reactions occurring in the body. The total chemical reactions occurring in the body. Metabolism. Metabolism can be of two types. We have two type of metabolism. One is called as anabolism. Another is called as catabolism. Anabolism and catabolism. Anabolism is when we make a complex compound. We use some simple compounds to make a larger complex compound, like photosynthesis. Okay, what happens in photosynthesis, Anna?
Energy is made. ATP is created.
Mm, yes, ATP is also generated, and what else?
Respiration.
Mm, photosynthesis. It's a process by which plants make their food.
Yeah.
Mm. So here plants will take out simple components and then form larger components. Clear? Yes. Okay, so that's anabolism. Catabolism is when we break down complex component. When we break down complex component and small form into smaller components. This is called as catabolism. So all this anabolism, catabolism, okay, as total as metabolism, everything is taking place in all of our cell. Okay, and the sole purpose of this is to generate certain compounds. Okay, generate certain compounds that will be used by our body and also generate ATP or energy that can be also used by our body. Okay, so these two are the important conclusion of why metabolism occurs in our body. Okay, now see, when a particular cell is there, a particular cell, this cell and this is another cell, these two cells are growing. Okay, they are growing, they are becoming larger. Why? Because in both these cells we find metabolism. Metabolism will lead to the formation of compounds, certain substances which will be collected in the cell. Now you see, as the cell grow, as the cell carries out metabolism, both the cell will grow in such a way that a point will come when they touch each other. I am talking about animal cell. When they touch each other, the moment these two cells touch each other, it means that, okay, it means that the growth of the cell is completed. Okay, it means that the growth of the cell is what? Growth of the cell is completed. Okay, growth of the cell is completed. Why? Because the two cells have touched each other. This phenomena where the two cells touch each other, and when they touch each other, here it tells us that the growth is completed. This is called as contact inhibition. Contact inhibition. This is an important property of the cell. Okay, it's an important property of the cell. It tells us when the cell is matured and it is no longer going to grow. That means it has grown the maximally it has grown. Okay, now we have a large number of animal cells. We have large number of plant cells. Okay, animal cell, plant cells. How did we see or how did we study so much of their structure? Well, of course, the microscopic organisms we were able to see and study their structure because of the development of microscope. Sana, can you define me what is a microscope?
It's a device
Yes.
that is used to um,
What it'll do?
see things that you can't see with your naked eye.
Yes. With the help of microscope, we are able to see microscopic objects, right?
Mhm.
We are able to see microscopic objects. So microscopic objects, that means what is the microscope doing? The microscope is magnifying the small image, it is magnifying and making it larger for us so that we can see the different structures properly. Okay, so that we can see what different structures properly. Is this clear?
Yeah.
Yeah. Okay, now let's see here. Okay, let me just go back once. Plant cell. Okay, fine. All right, so we'll come here. Okay, so microscope helped us to enhance and observe the various features of a cell. We have different type of microscope. We have compound microscope, we have scanning electron microscope. Okay, we have inverted microscope, different type of microscope, electron microscope. Different type of microscopes will help us to see the image. Importantly is that microscope will function, they function similar to our eye. In our eye, when the light falls on the retina, a real inverted image is formed. In the microscope also, a real inverted image is formed. Microscope will have, it will have a system, it will have a system of lenses, and these system of lenses will magnify magnify the image, magnify the image that we want to see, magnify the image that we want to see. Okay, now when we are talking about magnifying the image, so here magnification power is an important property of the microscope. Magnification power, that means how much bigger the microscope can show that small object. Magnification power.
Mhm.
Okay, so for that we use many type of lenses. We use 45 objective, we use 10 objective, we also use oil immersion 100x magnification, okay, in compound microscope. Okay, the most advanced will be the scanning electron microscope. So various microscopes will help us to see the various structures in a more better manner. This is an important fact that we need to understand. Observing the cell, studying the various structure of the cell would not have been possible if the microscopes would not have been developed each time the cells were observed for different organelles. Okay, now how do we measure? You know, sometimes we can even measure in the microscope by using certain scales. Okay, by using certain microscopic scales. Okay, we use them, okay, in the microscope to measure the I mean the length, or measure the give a measurement of the cells that we are observing. Okay, lot of microscopes are there, they help in different manner and help us to observe the different different characteristics of different cells. Okay, this is clear, Sana? Any doubt here?
No.
No. Okay, all right, so let's see there. We have seen about the cell wall, so let's see how the magnification has helped us to study the eukaryotic cell as well. We have seen the prokaryotic cell. Okay, now we are going to see the eukaryotic cell. Let us do the eukaryotic cell in little bit detail with few of the structures. Okay, so all right. Okay, so we have the animal cell. The animal cell is delimited by always by a plasma membrane. Okay, plasma membrane. Then we have the cytoplasm. What is plasma membrane? Plasma membrane is semi permeable in nature. Plasma membrane is also selectively permeable. Selectively permeable. Plasma membrane is the outermost membrane of an animal cell. In the plant cell and in the bacteria, plasma membrane is present inside the other layers. Sana, can you tell me what is the difference between semi-permeable and selectively permeable?
Selectively permeable is like it can choose what it
Hmm.
wants to let in.
Yes. And
And
semi-permeable?
semi-permeable is like um
It's very easy, somewhat related to that.
only like certain substances.
Hmm. Semi-permeable means plasma membrane will allow, it will not allow all substances to pass through.
Mhm.
It will allow only some substances to go inside.
Yeah.
Now, selectively permeable, we have different cells in our body, and different cells will allow only different components to get inside. So different cells are selecting which compounds will enter into our cell. That is selectively permeable. Semi-permeable is a general nature of plasma membrane where it will not allow all substances to pass through. Clear?
Mhm.
Yes.
Yeah.
We'll study the plasma membrane structure. Before that, cytoplasm. This is colloidal. This is colloidal in nature. Cytoplasm also will carry out all kind of chemical reaction occurs in the cytoplasm. The cytoplasm is also that part of the cell where we will see different membrane bound organelles are suspended. For example, we will have mitochondria. Okay, let's see here. Mitochondria. This is what? Mitochondria. 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 This is called as the powerhouse. It is called as the powerhouse of the cell. The mitochondria in the higher form is involved in oxidative. It is involved in oxidative phosphorylation. Oxidative phosphorylation. Mitochondria, oxidative phosphorylation. That means it is the production. It means the production of ATP. Production of ATP. Okay, oxidative phosphorylation, production of ATP. What is ATP? It is the energy currency of the cell. Okay, you go to a imagine you go to a restaurant and you want to eat a garden burger, okay, or garden salad or burger, whatever. Okay, so you have to pay the restaurant person with the electronic money, or maybe you have a cash with you, right? Only once you pay, then only you're going to get your food. Am I right? Yes,
Yeah.
without paying, will the restaurant person give you food unless and until he's your friend, right? Yes.
Mhm.
And you often go to a friend's restaurant, right? Yes. I do go. Yes, we enjoy a lot in a friend's restaurant. We cook also there.
Cool.
Okay. All right. We have a lot of dishes, and I'll be introduce some new ones, and then we all friends have a get together at weekend, and then okay, all right. So what do we do? Suppose if I go to a restaurant, I order for a burger, and then what I do, I give the cash to the person, and then after having my food, I'll pay for that, for the food that I have, the services that they have provided me. I'm going to pay for that, right? Without paying, I cannot come out of the restaurant, or I have to pay. That's very important. Even our cell also needs payment. To perform any function, our cell needs energy. And that energy comes in the form of ATP, adenosine triphosphate, which is called as the energy currency of the cell. Clear? Energy currency of the cell. Okay. All right. Mitochondria. Now, next. Okay, so mitochondria. So we will see the structure of mitochondria, but first let us do the other cells also, other types. So we have here the entire cell is divided into compartments, and these compartments are called as endoplasmic endoplasmic reticulum. Okay, now endoplasmic reticulum originates from the outer nuclear membrane. See, this is the outer nuclear membrane. Outer nuclear outer nuclear membrane, and this is the inner nuclear membrane. Whatever the fluid is here, this is called as nucleoplasm. This is the fluid inside the nucleus. This is the fluid inside the nucleus. Fluid inside the nucleus. We also have chromatin fibers. These are nothing but what? They are chromatin. They are chromatin fibers. We also have a structure which is termed as nucleolus. Nucleolus. Other than that, in that cell, we will also have another structure which is termed as Golgi apparatus. Golgi apparatus. Okay, Golgi apparatus. Clear? Okay. All right. Okay, so these are some of the structures that will be found in the cell. We'll also find lysosomes, we'll find ribosomes. So we're going to study all of them. So right now you have a clear-cut view. Okay, you can observe, okay, what the animal cell or what are the contents of a eukaryotic cell is. Last class we did plant cell, which is also eukaryotic cell. Okay, so let us see some of the functions that you need to know here. Okay, so we go first with the mitochondria. The mitochondria are called as the powerhouse of the cell. The mitochondria are the ones which are involved in the energy production. Okay, energy production. This is the outer membrane of mitochondria. This is the inner membrane of mitochondria. Between the outer membrane and inner membrane, there is a space. This space is called as perimitochondrial space. At times the inner membrane is folded forward into the cytoplasm of the mitochondria is called as the matrix. The inner membrane is thrown into folds on the matrix. This fold is called as cristae. What is it called? Cristae. Okay, cristae is called. And it is this cristae is very important because whatever the ATP generation will take place in this area only, that is in the cristae. In the cristae we will have the generation of the ATP. ATP generation will take place in the cristae. Okay, inside the mitochondria, very, very important. Mitochondria have their own DNA. They have their own RNA. Okay, they have their own DNA and they have their own RNA. Even the ribosome will measure. Okay, ribosome will measure what? 70s ribosome. Mitochondria is a structure which is present in eukaryotic cell,
Mhm.
but its ribosome sedimentation coefficient is what? 70s. That is very surprising because 70s is similar to the rhizomes which are found in bacteria. But mitochondria is not found in bacteria. It is found in other organelles. Okay, in eukaryotic cell. We'll see why this is so. Okay, mitochondria. Okay, so they have oxidative phosphorylation. Mitochondria will carry out, okay, wherever there is mitochondria will carry out aerobic respiration. Okay, what it will carry out? Aerobic respiration. Okay, this thing you need to understand. This is clear, Sana? Any doubt?
No.
Okay, all right. See how the things are working. Okay, how the things are working. Now, concentrate. I'll tell you about this. Now, let us see. There is another organelle which is present, mitochondria. Okay, mitochondria. This is mitochondria. It is present in both plant and animal. Okay, it is called as powerhouse of the cell. Let's see now another structure which is found only in the plants. That is the chloroplast. Chloroplast is found only in where? Only in the plants. Okay, chloroplast. Within the cells in the higher plants, there is a discoid or disc-like structure. Okay, discoid or disc-like double membrane structure. Sana, have you studied before about the chloroplast
Not in detail.
structure? Yes, what have you studied? Tell me.
Not the structure of the
Hmm.
chloroplast.
What you have studied?
About the chloroplast?
Hmm.
Just its like what its like main function is.
It is the main, it is one of the most important structure. In chloroplast, there are pigments. In chloroplast, there are what? Pigments. And these pigments are called as
Chlorophyll?
chlorophyll. And they are the one that is involved in the process of photosynthesis. See here, what chlorophyll will do. Chlorophyll will write short form as CHL. Okay, you can also write it CHL. Chlorophyll will absorb light. Chlorophyll will take away this light, convert the light energy into chemical energy in the formation of new compounds. And this is the process of photosynthesis. Okay, so this is what chlorophyll does. And the structure that contains the chlorophyll in the plant is called as chloroplast. The chloroplast, you can see it is a discoid in shape. Discoid in shape. This is the common shape. Disc-like. Okay, disc-like. There is an outer membrane. Outer membrane, and we have inner membrane. See this nicely, you see, because in one of your syllabus, you also have to study about the endosymbiotic theory. That's why I'm showing you the structure of chloroplast. Okay, inside the chloroplast, there is a fluid filled space. This fluid filled space is called as the stroma. Okay, there are some grain like structure which is stacked one above the other. See this grain like structure which is stacked one above the other. Okay, and these structures are also connected. So let's see. In each of this coin, like, can you see this grain? Like, can you see this?
Yeah.
Yes, each of this structure, each of this structure is called as thylakoid. And it is in the thylakoid that the process of light reaction and light reaction of photosynthesis and ATP formation takes place. Okay,
Mm-hmm.
what is that? Light reaction of photosynthesis. Photosynthesis have two phases. One is dark reaction, another one is light reaction. So light reaction takes place where? Thylakoid. Okay, and ATP production. So all of this individual is called as a thylakoid. This coin-like structure stacked together, this is called as the grana. So I can even say that light reaction occurs in grana. This structure that joins two grana, this is another grana. This is called as the stroma lamellae. Okay, what is it called? Stroma lamellae. All right, okay. Now, here in the matrix, you'll find various enzymes. You'll find the deposition of starch materials. Okay, so many compounds will be there. Basically, this is a very simplified structure of chloroplast. If you know this, it's more than enough. And you should know their function. Where are the chlorophyll present? Chlorophyll will be present inside the thylakoid. Okay, and the function of chlorophyll molecule is to convert the light energy into chemical energy during the process of photosynthesis. Okay, during the process of photosynthesis. This is stroma. It is in the stroma that the dark reaction of photosynthesis will occur. Photosynthesis is divided in two phases, dark and light reaction. Okay, so light reaction, you need to study all this when you do photosynthesis. You have to study all this. Light reaction occurs in the thylakoid or in the grana. Dark reaction will occur in the stroma. What is stroma? It is the fluid matrix of the chloroplast. Okay, it is the fluid matrix of the chloroplast. This point is clear?
Mhm.
Okay. Chloroplasts have their ribosomes. Okay, now you see this point. Now try to see this point. So both in mitochondria, and in chloroplast, both in mitochondria and chloroplast. Okay, we have 70S ribosome. Okay, the sedimentation of ribosome is what S? 70S. Now see, Sana, they are present in 70S. 70S ribosome is a type of ribosome which is present in prokaryotic cell, right?
Yeah.
So what happens here? The ribosomes. So how come mitochondria and chloroplasts which are present in eukaryotic cell, they have 70S ribosome of prokaryotic cell? Can you tell me how is it possible?
I'm not sure.
Yes. See, this is a eukaryotic cell. Inside eukaryotic cell, we have mitochondria and chloroplast. It is 70S. But the eukaryotic cell as a whole is 80S. Okay. All right. The reason is a theory which is called as the endosymbiotic theory. According to endosymbiotic theory, what happened? Okay, the endosymbiotic theory tells us how the eukaryotic cell originated. They engulf the two cells which is similar to, okay, two cells, one cell is similar to mitochondria and the other cell is similar to chloroplast. So this larger cell, what they did, engulfed, okay, engulfed both the cells and kept both the cells with it rather than destroying it. Okay, that's how we get mitochondria and chloroplast inside eukaryotic cell. Okay, did you understand?
Mhm.
Yes, yeah. Can you explain me what you understood? Yes,
I'm sorry, can you repeat it?
yes. That's what I asked you. See, I'll tell you a story. Listen to the story. Okay, all right. Once there was one cell. Okay, once there was one cell. So we have this cell, one cell. Okay, all right. There was another cell. Okay, there was two cells were there. They're prokaryotic cells. They're prokaryotic cells. Okay, all right. We'll see now what they do. Okay, this cell, what it did was it trapped light energy, and with the help of light energy, it started to produce food. Whereas this cell, what it did was it would use the food and break down the food to release ATP or energy. Till here, the point is clear.
Yeah.
Okay. Now, they were doing their functions. They are prokaryotic cell. One day, a larger cell came, and this large cell was so hungry. Okay, it was so hungry. It says, I'm going to eat everything I get. Okay, it says that I'm going to eat everything I get, whatever I get. I'm too hungry, and I've been hungry for so many years. Okay, so what it does is that it turns its head and it sees light. Okay, sees this cell, which is producing what?
Food.
Which is producing food. Okay, all right. Then it turns its head towards the side. What does it see?
ATP.
Yes, the cell is producing ATP. It says, wow, I'm going to eat it. And he does that. He eats them both. Okay, he eats them both. Once he eats them, after that, what happens? These cells, they are inside his stomach now. Okay, they're inside his stomach. See this red color, this yellow color. Are you understanding?
Mm-hmm.
Yeah. Okay. So red color, yellow color was producing what?
Food.
Food. Red color,
ATP.
ATP. Then this larger cell said, one is producing food, one is producing energy. If I eat them, then I will be in loss. At least they are making food for me and they are giving me energy. So this larger cell said, no problem. Let them stay with me only. Okay, let them stay with me. So in course of evolution, this larger cell, this larger cell is the eukaryotic cell. Eukaryotic cell. The smaller cell, okay, the one that produce ATP, this is mitochondria. The one that produces food, this is chloroplast. So now you see eukaryotic ribosomes. The ribosomes which are present in eukaryotic cell,
Mm-hmm.
ATS. But since chloroplast and mitochondria were long time back, were once prokaryote, that's why in their ribosome they are what? 70. Yes.
Mm.
Clear or not?
Yeah.
Yes. Okay, now you understood. This is
Yeah.
called as the endosymbiotic theory. Okay, it is called as the endosymbiotic theory.
Oh, Miss, I'm sorry. Can I go to the bathroom for a second?
Sure, why not?
Thank you. Hi, sorry.
That's no problem. That's not an issue. Absolutely fine. Yes. Shall we start? Yes.
Yeah.
Yes. So, Sana, did you understand what is endosymbiotic theory?
Yeah.
Yes. Okay. So now, so we studied about the mitochondria. We studied about chloroplast. We studied endosymbiotic theory. Next, we shall study about some more organelles. Okay. Now, have a look at this. This is one organelle that we studied, endoplasmic reticulum. Can you see this?
Mm-hmm.
Yes, endoplasmic reticulum. So let's work on that. Endoplasmic reticulum. The endoplasmic reticulum, or the ER, we also called it. Okay, they extend from outer nuclear membrane, outer nuclear membrane to, okay, outer nuclear membrane up to the plasma membrane. Outer nuclear membrane up to the plasma membrane. They extend. Okay. Now you see them. Suppose this is the outer nuclear membrane. Okay, this outer nuclear membrane. So it is from here, then the endoplasmic reticulum will start and up to the plasma membrane. The endoplasmic reticulum, if you enlarge this area and you see, then these are made up of flattened structures like this. Okay, flat elongated structure. These flat elongated structure is called as cisternae. Okay, they are called as cisternae. Also, we will find tubular structures. like this. These are called as tubules. Then we may also find rounded structures called as vesicles. Vesicles inside them contain some liquid features, liquid component. This liquid is nothing but, or it's a solution, what the endoplasmic reticulum has made. Okay, so these are the three components. These are the three components: cisternae, tubules, and vesicles make up the endoplasmic reticulum. There are two type of endoplasmic reticulum. In one endoplasmic reticulum, okay, in one endoplasmic reticulum, what happens is, on the surface of the cisternae, there are granular structures, grain like structures. And when you observe them from the top, you'll find that these endoplasmic reticulum appear to be rough. And these grain like structures, okay, these grain like structures are nothing but they are ribosomes. Okay, and since there are ribosomes on the surface, so from the top it appears rough. So this endoplasmic reticulum is called as RER, that is rough, yes, endoplasmic reticulum. Okay, so can you tell me what is the function of the RER, rough endoplasmic reticulum?
Is it to create protein?
Yes, because it has ribosomes, and ribosomes carry out what? Protein
Yes, yeah.
synthesis, right?
Mm-hmm.
Yes. So RER will carry out protein synthesis. There is another one, rough endoplasmic reticulum, which does not have any ribosome. It is just smooth. This is called as the smooth ER. It has two functions. Smooth ER is involved in lipid synthesis. Lipid means fat. And also they carry out detoxification. Detoxification. Okay, detoxification will be carried out by the smooth endoplasmic reticulum. Keep this in mind. What is the function, Sana? What is the function of RER, rough ER?
To create protein.
Protein. What is the function of the smooth ER?
To
Yes. What is the function of the smooth ER?
Synthesize.
See, I have written there. See at the side, smooth ER is written. Below that, I've written
Lipid synthesis.
lipid synthesis and detoxification. Okay, detoxification is making the harmful toxic material less toxic and then eliminating out of their body. That is called as detoxification. This is smooth ER and rough ER. Then what happens after the protein is made? This protein will be filled in vesicles, and that will go for packaging to the next organelle, that is called as Golgi apparatus. Whatever it is, whether the endoplasmic reticulum is making protein or it's making lipid, okay, it's making protein or it's making lipid, what happened, both will be packed in vesicles and then sent to Golgi apparatus. Okay, then sent to Golgi apparatus. This point is clear? Yes. So whether protein or lipid, Both of them, which is made in the endoplasmic reticulum, they are just raw materials. Okay, suppose, for example, you want to drink juice. Okay, you want to drink juice. I want to drink apple juice, and you say, I want to drink apple juice, but I give you apples. How will you get juice from them?
You.
Yes, you want to drink apple juice,
Yeah.
but I give you only apples. I don't give you juice. I tell Sana, you have to extract the juice and you make your juice. You now you do. I don't. So what is what? How will you make?
But extract it.
You'll extract. Will be a juicer, right?
Yeah.
Yes or no?
Yeah.
Okay. Now you see. I gave you apple.
Mhm.
Apple. What you require? Apple juice. I give you apple. I am endoplasmic reticulum. Okay. I had apple. I gave you apple. Can you see that lipid and protein
Mhm.
in the vesicles? Those are apple. Okay. Then see that Golgi apparatus. Golgi
Mhm.
apparatus, can you see?
Yeah.
Yes. That is you. You take the apple. And what you do? You make what you do?
The juice.
Yes, you extract the juice.
Mhm.
And then what happens? Once you extract the juice, see here, I'll show you Golgi apparatus, how it will look like. See, like this. Golgi apparatus also, vesicle-like structure. See? Okay, like this. So what you do? Okay, I take the vesicles here, all protein and lipid vesicle, from one to another. Finally, what you're doing? You're extracting the juice. Finally, juice is ready, right?
Yeah.
Yes. Now, how will you drink the juice? Will you drink it in the juicer itself?
No.
No, what will you do?
I put it in a glass.
Yes, you put it in a glass. You now from the Golgi apparatus, another vesicle will come out. This is the apple juice. All the components. What the endoplasmic reticulum had sent to the Golgi apparatus have modified it and packed into vesicles. Okay, this you get your apple juice in the glass, and if you want to drink it, you can drink it, or you can serve it to others. Right?
Yeah.
In that way, when we need proteins and lipids, endoplasmic reticulum will make it in the raw form. Then it sends into the vesicles, into Golgi apparatus. Golgi apparatus will modify it. The function of the Golgi apparatus is to carry out packaging, packaging of materials, and then packaging of materials, pack them into complete form. Later, this can be used by the cell. This point is clear.
Yeah.
You understood how things are made? Yes.
Yeah.
Now see this. So the position of the Golgi apparatus. See where the Golgi apparatus is present. Whatever vesicles are made by the endoplasmic reticulum, this will go to the Golgi apparatus. Golgi apparatus will make them in vesicles and send them to different parts of the cell. Clear?
Mhm.
So now, understanding or not?
Yeah. Understanding.
Yeah. Okay. All
Mhm.
right. This is Golgi apparatus, rough endoplasmic reticulum functioning. In the Golgi apparatus, there will be another organelle that will be formed. And this organelle is a very typical organelle. This is called as lysosome. Inside it contains a fluid, and that is nothing but acid hydrolases. Acid hydrolases, or I can say hydrolytic enzyme. hydrolytic enzyme. Acid hydrolases or hydrolytic enzyme. Lysosomes. Lysosomes in general term, sometimes we call them as suicide bags of the cell as well. Okay, they will digest the cell. See why they're called as suicide bags. Okay, why they're called as suicide bags. See, this is very, very simple. A cell is old. Okay, our cell divides, they grow old and they get destroyed. Now the cell is old. The cell has to be destroyed. So we have a cell here, we have a nucleus here, and we have this lysosome. There are many lysosomes. So when the cell grows old, all the lysosomes will combine to form a bigger lysosome. Okay, all of them will combine to form a bigger lysosome. And a time will come when the lysosome will what? It will burst. Lysosome will burst. Okay, and when it bursts, it releases the acid hydrolases, hydrolytic enzymes into the cell. An entire cell will be destroyed. That is why it is termed, sometimes in some books you'll find it is termed as the suicide bags of the cell as well. Okay, and it is the place, it is that organelle which will result in cell death. This point is clear?
Yeah.
Yeah, understood. Okay. All right. Okay, so this is lysosome. So we have studied about Golgi apparatus. We have studied about lysosomes. Sometimes you'll find vesicles, small vesicles with all the components. Okay, then another organelle that will be there, which is called as, I'll write it here, which is called as ribosome. The sole function of the ribosome is to carry out protein synthesis. It's to carry out what? Protein synthesis. So protein synthesis. Okay. So ribosome structure is like this. It has a larger subunit and smaller subunit. This is larger subunit. This is smaller subunit. This point is clear?
Yeah.
Larger subunit, smaller subunit. Both the subunit are kept together by magnesium ion. This is the structure of ribosome. Ribosome do not have any membrane. Do not have any what? Membrane. So what is the most important function of ribosome, Sana?
They protein synthesis.
Very good. Protein synthesis. Okay. We have studied all these organelles. When we do mitosis, we'll study some more. Okay. Now what we will do, we will study the process. We will study the plasma membrane. You know mitosis, meiosis? Yes?
Yeah.
Yeah. Okay. So when we study that, we'll study those. Now we will study the plasma membrane, which is very important. Okay. So Sana, till now, have you got any doubt? Have you understood or not?
Yeah.
Okay. Let us see. Plasma membrane. Can you tell me, Sana, where is plasma membrane found?
Surrounding the cell.
Sorry?
Surrounding the cell.
Yes, surrounding the cell. Is it found in plants only, or is it found in plant, bacteria, everywhere?
Everywhere.
Yes, everywhere. Plasma membrane is a structure, okay, which is present in both plant cells and animal cells, including bacteria. Okay. Plasma membrane, very important. Plasma membrane. The plasma membrane structure, this structure, the bilipid layer structure, the plasma membrane, okay, the structure was given by Singer and Singer and Nicholson. They gave a theory called as the fluid mosaic theory. According to that theory, we have to study the plasma membrane and let's see the different features. Our plasma membrane has bilipid layer. Bilipid layer. Bilipid layer. So this is one lipid, this is another lipid. See? Okay, see here how the lipid layers are attached. Okay. All right, I'll just draw this and then you'll understand. Okay, bilipid. This is the bilipid layer. Bilipid layer. Okay, bilipid layer. Now, the lipid that is present in the plasma membrane is phospholipid. Very important MCQ. Okay, multiple choice question. Phospholipid. So this phospholipid has a head and a tail. See, if this is the plasma membrane, so this obviously would be the cytoplasm, and this is the extracellular fluid. See, this is our cell. Okay, so I have taken a section of plasma membrane, this one. So this is cytoplasm, this is extracellular fluid. Okay, extracellular fluid. This is cytoplasm. Now you see here. Okay, now try to understand. Now try to understand this. See. Okay, the in between the bilipid layer. Now you see here. This is the head of the lipid. Where it is facing? Towards the outside or towards inside?
Outside.
Outside. What is there outside? Whether extracellular fluid or cytoplasm. Is water present there?
Yes.
Yes, water is present there. So, since the head is facing towards the water component, the head is hydrophilic. The tail is inside. Hydrophilic is water loving. Tail, hydrophobic. Water repellent. What is water repellent?
It's water.
Yes.
Repulsive water.
Water repulsive, water hating. Yes or no?
Yeah.
Does not like water. So it is hydrophobic. Okay. Now we have here. Can you see this red color? Whatever I have drawn.
Yeah.
Yes. Can you tell me what are they?
Proteins.
Yes. They are what? They are proteins. Okay, they are proteins which are present inside the plasma membrane. Okay, types of proteins. So I'll color this. All right, so let's see. Okay, proteins. Some proteins will be present on the outside as well, like this. Okay, let's see now. Okay, so this protein, this is inside the plasma membrane, but half. It is called as intrinsic or internal protein. Similarly, this one, this is also called as intrinsic or internal protein. But since it is running all throughout the plasma membrane, this is called as transmembrane protein. Transmembrane protein. Okay, which can also be what? They can also be what protein? Carrier protein. Okay, and this is standing here. This is extrinsic, external protein present on the external surface.
Mhm.
Next, on the surface, we will have branch chain structures like this. Branch chain structures. These are nothing but, okay, these are nothing but they are glycolipid. Okay, they are glycolipid. All right.
Yeah.
Okay. Now, here they said that, okay, they use the word fluid mosaic. Fluid. That means the plasma membrane, or which part, the lipid layer, is constantly in movement. Okay, this fluid means, okay, fluid means that, okay, that the lipid layer is constantly moving. Okay, lipid layer is what? Constantly moving. Fluid. Mosaic. What do you understand, mosaic?
Um, they're like different
Yes, what do you
parts,
understand by mosaic?
like different parts put together.
Yes, mosaic. Sorry.
Like different parts put together.
Yes, different parts put together, but in such a way that you cannot make out that, these are different parts.
Yeah.
So when you see from the top, it appears like that, like a mosaic, like a pavement. So that's why they named it as fluid mosaic model. Now, I said that the lipid membrane is constantly in movement. That much movement is not good. So what we do is that, between the head of the lipid molecule, between the head like this, there are certain compounds. What these compounds will do is that these compounds, which are called as cholesterol, cholesterol will limit the movement of lipid layer. So if a question is asked, who controls the movement of the lipid layer, what your answer will be?
The
Who
phosphorus.
con- Cholesterol.
Yeah, cholesterol.
Cholesterol. Cholesterol. Cholesterol controls the movement of the lipid layer. Point is clear? Yes?
Yeah.
Yes or no?
Yeah.
Okay. So now look into the whiteboard. See the options there. See the components. You have any doubt? Have a look at that. Have a look at that. Any doubt? Anything you have not understood? Anything? See, need to understand here if you have to do well in your test. Yes or no?
Could you go through the glycolipids again?
Glycolipids. Okay. Glycolipids are present on the
Yeah.
surface of the plasma membrane. Okay, they're present on the surface of the plasma membrane. As you can see, they are branched. Can you see that? Yes,
Yeah.
they are branched, and their most important function is cell recognition. They'll help to recognize different cells. Okay, glycolipids, glyco, carbohydrate, lipid, fat. Whenever a carbohydrate is combined with lipid, it is called as glycolipid. Clear?
Yeah.
Yes,
Hmm.
glycolipids. Glycolipids. They are branched. Okay, they are made up when carbohydrate and lipid combine. They form glycolipid. Glycolipids, okay, they help in cell recognition. They are present on surface of plasma membrane towards the ECF. Okay, extracellular fluid. Extracellular fluid. ECF. What did I write in the last sentence? They are present towards the ECF. What does this mean?
Extra
Where they are present?
fluid
Towards the cytoplasm or towards the outer side of the cell?
towards the cytoplasm. No, the outside. The outer side.
Outside.
Yeah.
See, Sana, if it is inside the cytoplasm, how will cell recognition work?
Yeah.
Yes or no? Feeling sleepy? Yes. How will the cell recognition work? If it's inside, if you are hearing, if you are with your headphones and you're listening to music,
Oh no,
if I
I'm
call
not.
you, Sana, 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 listen. Will you listen? You'll not be able to hear. Yes or no?
Yeah.
Yes. If you want to hear my voice, you need to take out the headphones. Correct? Yes.
Yeah.
Same way. The cell recognition. Cell can be recognized only when the glycolipid is present on the outer, not towards the cytoplasm. Clear?
Yeah.
Okay. Yes. See,
Yeah.
anything else you have not understood? See, nicely, you see. I will repeat again. If you do not understood, I'll repeat again. You have a test. You need to do well in your test.
I think I understand.
Yes, understood.
Yeah.
Okay. All right. So let's see here. Now, see into this. Suppose, Sana, for example, okay. Suppose, for example, this is outside the cell. I'll draw another diagram here. Okay. All right. See here. This is transmembrane protein. Okay. Next. All right. I'll just draw a few only. That's it. Okay. Let's apply lipid layer. Okay. I'm drawing it again. Now see. Suppose, see, I'll just color this so that you can understand what I'm trying to say. If I leave it blank, the writings may merge. So when you see the PDF, you may get confused. Okay. So now you see. Suppose, so this is what layer? Bilipid layer. Lipid means fat. This is transmembrane. Transmembrane protein. Correct?
Yeah.
Yes. Transmembrane protein. Now you see here. I have here a compound which is soluble in water. I have a compound. Okay, I'll draw here. I have a compound. This is what? Soluble in what? Soluble in what, Sana?
Water.
Water. Okay, so this is a compound which is soluble in water. This compound tries to enter the cell through this region. but it fails. It utterly fails because this is a water soluble compound. And bilipid layer, this is made up of what? Fat. Can a water dissolve in in fat? Or can a fat dissolve in water? If you take a glass, put some oil, what do you see?
The
If you
oil,
take a glass of water, put some oil, what do you see?
oil floats.
Yes, yes, it floats. Correct. It floats. Similarly, this is a water soluble compound. Okay, that means it contains water. So, lipid, this fat, this lipid will not allow this to enter. Now, suppose this is the cytoplasm, and this compound is necessary for the development of the cell. So, who will allow? Who will allow? Okay, this water soluble compound to enter. Let us see. So, this water soluble compound comes here. What is this? What is this?
Transmembrane protein.
Protein. Transmembrane protein. So transmembrane protein, what it will do? It will allow the transport of water soluble components from outside of the cell to the inside. This transport, this transport of water soluble component, okay, this transport of water soluble component has happened through the protein. So this transport is called as facilitated diffusion. What is it called?
Facilitated diffusion.
Diffusion. Have you heard about that?
Yeah.
Yes. Now you know what is facilitated diffusion? Okay. Suppose this is point number one. Point number two. Point number two, I have water molecule. Can water molecule enter through the lipid?
No.
Can water molecule enter through the lipid?
No.
No. So how water molecule will enter?
To the
How water molecule will enter?
the transmembrane protein.
Transmembrane proteins, no. Water have another method. Okay. In our cell membrane, there will be a pore like this. There is a pore. You understand a pore, Sana?
Mm-hmm.
1 minute. There is a pore like this. Okay. There will be a pore like this, running throughout the plasma membrane. Okay, this is a pore like this. Okay, and this pore is called as aqua. 1 second. It is called as aquaporin. So aquaporin will allow the passage of whom? Water. Did you understand?
Yeah.
Yes. Now tell me the third substance, which is a fat, which is a lipid, from where it will enter?
If it's a fat.
From where it will enter? The lipid molecule. If there is a fat soluble molecule.
It will.
Yes, fat soluble molecule. Very simple, Sana. Fat soluble molecule.
In the.
Can it enter through the aquaporin?
No.
No, because water is there.
No.
Can it go through transmembrane protein?
No.
No, because there also water soluble is there. So which is left?
The
What is left in the diagram? We have
lipids,
used the transmembrane protein. We have used the aquaporin. Which is left?
the the lipids.
Lipids. Yes. Okay. So the last one will be water soluble, the fat soluble molecule will directly enter through the lipids. Okay. Fat soluble molecule. Clear?
Yeah.
Yes, this is how our plasma membrane will function. Okay, this is how the different plasma membrane functions. Understood or not? Yes?
Yeah.
Yeah. Okay. All right. So now, Sana, can you tell me what do you understand by the process of diffusion?
Um, it's the process in which um a substance goes from high concentration to a lower.
Higher to
Yeah.
lower concentration. Very good. Okay, from higher. So here it is movement of solute. From where? From higher concentration to where? Lower concentration.
Hmm.
Right?
Yeah.
Higher to lower. Any energy is required for this?
No.
No. We do not require any energy. This is diffusion. And just now I showed you through transmembrane protein what diffusion it is.
Facilitated diffusion.
Diffusion. This, what I'm showing you on the whiteboard, is called a simple diffusion. The one that we did through transmembrane protein is facilitated diffusion. Both of them are diffusion and both of them do not require energy. Clear?
Yeah.
So if they don't require energy, that means it is what process?
Passive transfer.
Yes. Passive process. Clear?
Yeah.
Yeah. Okay. So have you understood today's class?
Yeah.
Yes, clear? Yes or no, Sana? Are you understanding?
Yeah.
Yeah, clear, all clear, seriously. Or
Yeah.
something you have not understood. You're simply saying, yes, I've understood.
No, I understand. If I don't and I like review and I don't understand, then I'll ask.
Okay, fine. That's absolutely correct. Okay. But you're understanding in the class, your point picking up, right? Yes. Okay. So I'll be sending out some homeworks. Just complete the homeworks and send them. Okay. All right. I'll send you some questions as well. Clear?
Yeah.
Okay. All right then. Shall we end the class? Yeah. Bye. Take care.
Bye.
Meet again next week. Bye.
Bye.
Bye.